Okay, so I think we were looking at the biology part, DNA storage. Gave you some ideas. I will go briefly about one topic again, which is central dogma of biology, and try to see how it works. After that, uh, I'm going to give you certain other bio operations. You know, what are the different bio operations that you can do with the DNA, and then plus we are going to look at two important uh, techniques. One is PCR, another one is gel electrophoresis, right? So, what are these things? So let me start. So we were looking at this history of biology. We looked at different life forms and story of ships last time. Now we were looking at this process. This is central dogma of biology. That whenever our body has a special requirement for some protein, so what it does in the genome, look at different chromosomes. There are different chromosomes. Okay, you look at a chromosome. Okay, look at the specific DNA. And then you can read that DNA, and that DNA is converted. So there are different ways you can read the DNA. So when scientists are trying to really understand, so where is the information actually, for making protein. So if you take a string of say length n, okay, so you can start reading the genome from first bit of ACGT and then see what happens, or read it from the second bit and then see what happens, or read it from the third bit and see what happens. This is one direction. So one string of ACGT will correspond to 3 reading frames. Are you getting my point? 3 reading frames. You can start reading from the first bit, or leave first bit and start reading from the second bit, or leave second bit and start reading from 3 bits. Why 3? Because we know that this genome, this string of ACGT is mapped into, so each codon means 3 bits you read and converts into an amino acid through a map. So that's why 3 bits are important. Now 3 bits, if I'm asking 3 bits, should I start from the first bit or the second bit or the third bit? From where we should start? Okay, so that's why given a DNA string, it is making protein, it could be starting from first reading frame, second reading frame, or third reading frame. Now, this is in one direction. You can also look from the other direction. So, essentially, given a DNA string, you have 6 reading frames, 3 in this direction and 3 in the opposite direction. Okay. So now, out of these 6 reading frames, then you see where is the start codon and where is the stop codon. Because in the genetic code, there is one start codon and there are 3 stop codons. Each codon is made of 3 bits of ACGT, right? Or ACGU, whatever you call, if it is RNA, right? So basically, when scientists are trying to discover, so they do this kind of analysis. Take a genome string and then you try to map it and see where you can get a protein, actually, right? So reading from this way or this way. So I think if you look at one of our software, biospectrogram, which was developed by Neelay Chheda and Naman Turakia. So in this software, you can actually get the genome directly from the NCBI server. And then what we do, the software allows certain numerical sequences. So basically, because now you have got the data in the form of ACGT, now you convert this data into numerical sequences. There are different kind of maps possible. At least 16 different maps are implemented in the software if you look at biospectrogram. So what you do, so one map could be, so wherever there is A, you put 1, otherwise put 0. So you get a binary sequence, right? Similarly, you will go for C. So characteristic sequence for A, characteristic sequence for C, characteristic sequence for G, characteristic sequence for T. So each, so you will get binary sequences, right? So basically, Then you apply some sort of transform. It could be Fourier transform or something. So actually, what has been observed in what is known as genomic signal processing. So you know DSP, digital signal processing is very common, but in the genomic signal processing, what happens? You take the data, convert it into the numerical, and if you look at the power spectrum, whenever there is instructions for making protein, there is a peak in the signal. Okay, so this way you can identify that there is a protein. Okay, so this software that we have developed, there is a window of certain size, and it moves along the genome on one direction, on the other direction, and see where is a protein. Okay, so that is just one characteristic. Similarly, since I mentioned any genome, in the human genome, there are only 2% of genome which codes for proteins. And there are other characteristic things are possible. For example, RNA. So whenever a central dogma is happening, so DNA to RNA, RNA to biology, so RNA to, protein. So what happens? Many times from DNA, only RNA is produced and no further protein is produced, only up to RNA. So that RNA is basically used as a catalyst for certain reactions. So they are known as non-coding RNA. It's coding RNA which are coding for proteins, and non-coding RNAs which are not coding for proteins, but they are used as a catalyst in certain biological processes, right? So same genome, but so many possibilities. It's really beautiful, right? How all this information is arranged in the DNA. Right, so whatever you want. So sometimes you want a specific RNA to be produced, right? So body knows from where to start reading and where to stop. Okay, start reading is there is this start codon. You see from where the start codon starts, and then there are different possibilities of stop codon, right? So you can read the portion. This is how you read the genome. Now, there are plenty of software. There is for one very popular software is GeneScan, which can do this. So people are actually using certain probabilistic concepts, hypothesis testing, for example, to actually see where is a meaningful information in the genome, right? So they have observed. So they assign some sort of probability distribution and see whether it is matching with that distribution or not, right? If it is matching or differing, all kind of experiments people are doing. So actually, you can do a lot of dry lab experiments with a computer. You can try to understand the genome, right? So plenty of species are there, and we have very little understanding about the data. Data is available, and you can actually try to do. I should just mention another interesting work that we have done was classification of RNA secondary structures. So whenever there is a new virus coming, you can actually try to match it with the unknown virus in terms of the structures. So basically, if you can classify RNA. So there was a paper by one professor that, RNA secondary structures can be classified using graphs. They associated a graph from the RNA secondary structure, and the graph classification gives you the classification. It's a very, very naive approach, may not be very accurate, but it does give you some results, So we have also done some work. I think that paper, you can look at it. That paper is RNA as a permutation. You look at each RNA as a permutation in the group theory that you see. Okay, and how you can actually do a classification. So actually, beautiful algebra you can apply. In terms of algebra, I should say that when a protein interacts with another protein, or when a protein interacts with another small molecule, these interactions can be described in a very beautiful algebra known as Clifford algebra. Okay. So Clifford algebra is very useful. It is also useful in quantum computing. It is also useful in wireless communication. And it is also useful in biology, right? So ultimately, beautiful mathematical concept. You can apply it different places. Okay, that is just one thing. So okay, so here we were looking at. So now I'll do an example for you. Let me see if you can really understand. Okay. So I think I can just say that we have these alphabets, right? So I call it as sigma DNA, and this is sigma RNA. This is sigma AA, amino acids. Right? So then we have a star here, a star here, a star here. So it means the star means strings of any size, right? So suppose I take A, C, C, G, T, D, C. This is our X DNA, right? Belongs to sigma DNA star, right? I've taken some string. Then what happens? Actually, now this is converted into RNA. So T will be replaced with U in the first process of the central dogma. As you can see that here, first step is DNA to RNA transcription. So you get xRNA, right? So which is something like A, C, C, G, U, U, C. Right? This is the thing. So now we have got the RNA string. Now what to do? We have to read 3 bits and assign a protein. But you cannot assign anywhere. Actually, it has to go from the start codon. So we need to look at the start codon. So if you look at the table here, look at this table here. So see here, this gentleman here sitting, is the start codon. Okay, and there are 3 stop codons. One is here, another one is here, another one is here. So what is the start codon? AUG. Which stands for bismethylene, right? So AUG corresponds to this amino acid. Okay? AUG corresponds to this amino acid. And then the stop codon should be there, either UAA. So any gene ends with either, any gene starts with MET and ends with either UAA or UAG or UGA. Okay? So this is the thing, the start codon, stop codon. Okay? And the instructions for making proteins between the start codon and stop codon. Okay? So now, obviously, when you look at here, so you need to see where is that. So what is the start codon? I think I can again just recall. AUG, right? AUG is there. AUG is your start codon. So wherever there is AUG in this string, can you see anywhere AUG? You don't see it here, right? So, I mean, you cannot do anything here, right? Another observation. So basically, if you take some DNA sample, right, and suppose there is a stop codon in between you observed, that means again there is a issue. That means that is not a gene. You understand that? Gene has to be a start codon and stop codon in between the string, right? So if there is a stop codon, so you just take random sample of DNA, right? If there is a stop codon in between, so then certainly this is not the gene, right? It has to go from, and I am talking about all the reading frames actually. Okay. So anyway, so that means of course we cannot do, but of course if you want, you can just mathematically you can combine ACC from the table where it will go, and GUU where it will go, and so on, right? You can do all that, right? So another important thing I would like to tell you here, if you look at this genetic code, each amino acid has different encoding. For example, leucine, LEU. LEU, how many? So those, one is UUA, UUG, CUU, CUC, CUA, CUG. So in some species, one particular codon is used. Maybe in humans, if this amino acid is used in building a protein, right? So maybe I don't know exactly, but what I'm saying that one codon is used for humans. another codon for the same amino acid in a protein, some other codon is used. What is the reason? Why different codons are used for different species for the same protein, same amino acid? So this gives rise to a very interesting problem known as codon optimization. Scientists are always trying to do the engineering things, right? So for example, they want to see that they have observed there are certain crops which, these are seasonal crops, right? So they can happen in a particular season, right? So if you want to grow them in some other season, basically you can just change the codons in the amino acids. They are trying to observe, right? So basically you can play with the nature, right? You change the codon and see what happens, and they are able to do certain successful experiments that, okay, something beyond what is possible, you can do that, right? So there are many companies which are having, in fact, we have written a software long time ago on codon optimization. So now you can optimize the codons. There is a codon table, there is a server in Japan which lists different codons possibilities for different species, right? So this genetic code is kind of universal. There are other kind of genetic codes as well, but this is the most common one. So basically, the frequency of occurring these codons in each species is different actually, right? So which one is used for which particular protein or which amino acid is a very interesting thing, right? We need to understand this idea, right? Why this is happening, why a particular specific codon is used in a particular amino acid in a particular protein. Because protein is made of amino acids. Protein is nothing but chain of amino acids, right? So leucine is used in one amino acid, another leucine is used in one protein, same protein in another species. The different codon is used for the leucine. Here the different codon is used for the leucine, right? So that's a understanding that we want to do, right? So many interesting questions have come up that, so so this is really beautiful. So this problem is known as codon optimization problem. I'm sure if you search on Google, you will find many interesting things. So this is common, especially this was done with plants, especially in the botany department, people used to study these things. Why, especially in different plants, same protein, same amino acid, but different codon is used. There must be some reason by the nature, right? What are those things? So that is something interesting thing I would like to tell you. Now, look at this example. And can you tell me, can you just write down all the reading frames? The example is, so this mRNA has come. For your help, this table is there. The first reading frame will be what? If you read, so look at what is the size of this string? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 23, 20, 25 or something, right? How many? Size of this RNA string is how much? You are young people, right? You can read very. 24? 25? Huh? 24? Fine. So now what? I have to look at the reading frames. So the one way of reading frame will be what? I start from the first bit. So reading frame 1 in this direction. Okay. In one direction is UUG, then GAG, then UAA, then AAU. So can you fill the corresponding amino acids? UUG is what from here? UUG. See, UUG is leucine, LEU. First corner, I think I can just mark it. So when I look at this first reading frame, UUG, so U, U, and G. So this is the first one, right? Now the next second one, GAG. What will be the GAG? GAG is this one, ARG, right? Huh? GLU. G A GLU, right? Correct. Okay, sorry. So GAG is GLU, right? Because it's the last one, this side. Okay. Huh? A stop codon is there in the whole thing. So A is a stop codon, yes. Somewhere it is there or not. So as an exercise, I'm just telling you, you can write down the reading frame 1. Now, how to start the reading frame 2? I hope it is easy for you to write down the reading frame 1, right? Just take 3 bits, map out the corresponding amino acid, and you will have the reading frame 1. Now, when we go to the second frame, we can start from here. UGG is the first thing. So UGG is what? UGG. TRP. This will come here. Second reading frame, right? And like that, you can go on, right? Similarly, the third reading frame, right? Third reading frame will be from GGA. Got it? So 3 reading frame this side, 3 reading frame this side. Total 6 reading frames for a given DNA. Because we don't know from where actually you will find start and stop. But nature knows, right? If you have to go for this protein, just go into the DNA, look at reading frame from which position, and then you start producing the protein. Don't you think it's a beautiful thing, right? The whole system is so nicely arranged, right? Produce a particular protein. So, by the way, I have taken this picture from this exe file, which is there in lecture folder. I hope you installed that software. There are a couple of more exercises, so you can enter your amino acids and you can check the answer. It will tell you it's wrong or correct about your understanding, right? Okay. So I hope now it is much more clear. There are many other things. So I told you, right? Okay, DNA strand. You are getting, you're reading from single DNA to RNA, but actually it may happen. Splicing also happens. What happens? You read a portion of DNA from one chromosome, then another portion from another place, another portion from another place, and do the splicing. So mRNA string is a spliced string of all the strings together, and then you produce a protein. Also there, you see. Such a beautiful arrangement of the information, right? So whenever nature wants to produce a specific protein, I mean, it knows where to go and what to do. For example, you see, when a virus infects inside your body, what happens? How the immune system responds? The immune system, there are T cells and B cells, right? Right. And if you have to stop the virus infecting you, there are 2 things that you have to do. First, you should. So because you see, any virus, there are peaks, right? There is a receptor on the surface of the virus, right? Different receptors, and they are beautifully arranged, of course. Mathematically, they are very beautiful. Now, what happens? You need to somehow, because if this receptor gets access to your receptor on the other cell, right? So they will fuse, and the virus will infect inside the body, right? In that particular cell. So what happens? The body knows the antibodies are produced, which will actually attach to this, this thing, that receptor on the virus. So if you block these receptors, so bodies know, so our body knows where is the encoding for that thing, so that it can block that portion. Okay, it will block it. Block it means now the replication of the virus is reduced because it is not replicating, not infecting. Once that is reduced, there are other kind of cells which will digest this virus. Okay, so the whole thing is gone. And so there is a beautiful fighting between immune system and any foreign invader all the time. Every time there is an infection to you, this process happens inside your body without we were knowing, right? So when we take medicines, we are giving additional support to the immune system, right? Ultimately, it's our immune system which has to do the job. Okay. So Another interesting thing, so for example, if you suppose you got normal cold and cough, so you know that each virus has a specific entry point inside the body. So cold, cold and cough virus can, I have something, right? So can enter inside your body from this place, inside your talu or whatever you call it, right? So at that point, the cell receptor is which can common cold virus can access that receptor, and so it will enter the body. That's why they say that you would drink hot water or hot tea to the concentration of virus, because normally these viruses will die at the temperature of 32 degrees centigrade. So if you have a cold, if you take hot water or tea or something or whatever different kind of kadha, right? It will reduce the concentration. If the concentration is reduced, body can fight back easily. With the immune system will fight it and you are relaxed, right? So I hope now you know so many beautiful things about biology that you should probably try to understand on yourself. Anyway, so now I already talked about HIV. Now I have to cover certain bio operations on DNA. So the first one is, of course, this is, so you know that DNA is a double helical structure. That means cytosine, guanine, thymine, and there is sugar phosphate backbone at the back end from where these things are attached. So information is in the form of these base pairs, right? These base pairs are having information or instructions for making protein, and normally it is in the double helical. I mean, also this double helical is arranged in the form of a coil, around certain other specific proteins, histones, and so on. I mean, there also there are changes in terms of epigenetic changes happens. I gave you the example of epigenetics earlier, right? So in protein also those kind of recordings are happening anyway. So so two very important operations that you can do is melting and annealing. You can actually take a double stranded DNA, so ACGT, and then you heat it, increase the temperature. So what will happen? They will be separated. So hydrogen bonds between A and T and C and G will be separated. I think I earlier asked you how many hydrogen bonds between C and G and A and T. Which one has more? Any idea? Yes. C and G have 3 hydrogen bonds and T has 2 hydrogen bonds, right? So yeah. So basically you see that to break the bond in C and G require more temperature, right? And A and T is, if there are more things in A, that's why, you go for higher C and G frequency, when we want to fix the GC content. Anyway, so okay, so this is one. Second, when you decrease the temperature, annealing will happen, and again hydrogen bond is formed. And DNA computing is essentially based on this DNA hybridization, and this DNA hybridization is also the source of error. So that is also there. Okay, so this is melting and annealing, two basic operations that you can do. Cutting. So you can cut DNA at a specific position. As I mentioned earlier, it's known as restriction enzyme. Restriction enzyme is a specific kind of protein which can cut the DNA at a specific location. Okay, it acts as a Caesar. So for example, if this string is there, so restriction enzyme will find the restriction site on the DNA and it will cut it. So you can see that enzyme has cut. So there is a cut. So DNA cutting is possible through restriction enzymes. There are many restriction enzymes that has been identified. Okay, and so they are used for cutting the DNA at a specific location. Okay, restriction enzymes. DNA ligation. So ligation or DNA ligating is what? So this is again an enzyme for sealing the phosphate backbone. So if there are two DNA strings, or double strings with certain free hands, when you attach them, so hydrogen bond is done, right? But then the top A, and A here at the bottom will be free, right? To seal that, the ligation technique will use, to seal that phosphate backbone, there is an enzyme which can seal that. Okay, and of course this it happens in the 3D way, but understanding to seal the topmost thing, right? You require DNA ligation, right? So it's an enzyme which can do that. Okay. All these enzymes are proteins. So basically, you know, they do all kind of things, right? And also there is polymerase. I'm going to talk about it. Now synthesis, as I mentioned, I am going to talk more about it, the history, how it started, and so on. And what are the three important kind of, three important kind of synthesis techniques which are there. So we have free ACGT, right? And then basically what you can do, you can combine them to a longer strands. So this is kind of writing the DNA by using the chemistry. Okay. So we'll talk about that chemistry after some time. How this, because this is very important for DNA storage. You know, you need to discover new kind of techniques for writing the DNA, for combining these letters ACGT together in a specific sequence, right? So that is, of course, machines are available which are doing these things. Now, PCR. This is very beautiful discovery. I think I'm sure people are thankful to Gary Mullis in 1983. And this is, I can see them. So I was only 13-year-old at this point of time, right? When things were happening in biology, right? Just happening. First time, PCR was discovered. Suppose COVID has happened is this time without PCR, it would have been a walk, you know. You are not able to. So PCR is a technique to actually replicate the DNA, okay, or whatever the genome is. So basically, it stands for polymerase chain reaction. Polymerase is a molecule which will do the job. I will tell you how. So there is a statement in Scientific American beginning with a single molecule of the genetic material, DNA. The PCR, polymerase chain reaction, can generate 100 billion similar molecules in an afternoon, few hours. Okay, the reaction is easy to execute. It requires no more than a test tube, a few simple reagents, and a source of heat. Heat is good enough to give you photocopy. It's a photocopy machine basically. Any genetic material, DNA. So whenever, so why this is important? Whenever there is a, suppose crime has happened, right? And you get a small evidence of the DNA. Now that small DNA may not be sufficient to do all the experiments that you want to do. So you want to replicate it, many copies. So you do PCR. When you have enough data, take a small one and do one experiment. Okay, this is not there. Take another DNA and do another experiment. It's easy for you, right? So PCR is very, very important. Or even this viral infections. People first do PCR. Okay. And after that PCR, they get a signature of the virus, right? So it's a very, very important technique. So, and then this guy got Nobel Prize. I think I would, discovering a photocopy machine for the nature is really a great discovery. Right. And the idea is very simple actually. It involves the genetic material that you want to copy, the source. It involves primers. Primers are again short strand of DNA which are complementary to the start portion of the DNA from where you want to start, one side and the other side. Okay. So primer is a short strand of DNA which are complement to the respective sides of the genetic material. And then you require polymerase, which is going to connect the free ACGT materials into the whole thing. Let me show you the picture actually. By the way, you can do the PCR experiment virtually at this website by Georgia University of I think Utah, right? So this lab is there. Click here, and you can actually do PCR experiments. Go to the, you can do everything. Virtually pick up the test tube, put in thermocycler, and all kind of things. Put the reagents, put the primers, put the polymerase. So you can do without actually going to the biology lab. You can do the animated experiments virtually, right? So I invite you to just look at this and do it. Okay. So here is a PCR technique. What it has, you see, initially we have, this is our DNA template with sequence of interest. So what we do first, so we want to make a copy of this DNA. So what we do, there are three processes. denaturation, annealing, and elongation or extension. So basically, all are at different temperatures. You can see the first process is 94 to 96 degree. Annealing happens at 68 degree, and elongation happens at 72 degree. So 3 temperatures you have to use in each cycle. So what you do? What are the ingredients? You require the sample DNA, which is here, right? You require primers. So primers are complementary to this portion and that portion here. You see, these are the primers here. Okay, and then the free nucleotides, that you want to attach, ACGT should be sufficient number, should be there, and the polymerase, which is going to do this job. So what happens initially when you increase the temperature to 94, 96 degree, this hydrogen bonds will break and you get free strands of ACGT. 2 complementary, right? Then what you do? You reduce the temperature to 68 degrees centigrade. At this temperature, these primers will attach to the opposite side of the DNA. So these primers are complementary strings, small complementary strings of each side. Okay, at this side and this side. Once it attaches, again you increase the temperature to 72 degrees centigrade. Then what will happen? This polymerase will come into picture and will start at filling the this. You can see that here polymerase is attaching and making the whole complementary strand at the bottom, right? The first one and the second one at both the places actually. One primer is doing for this, another primer is doing it for this. Actually, primer design is very, very important engineering aspect. We want that primer should be error free. I mean, so again, sometime I can talk about it, how to design primers efficiently. So in biological process, primers are very, so it's a short strand of DNA which will start the whole process. That's why I call it primer. Okay? So they are opposite here, right? You can see that. So now these free strands will come and then you have a replication. So first cycle, so you started with one strand of DNA, I mean, this double-stranded DNA, and you have 2 copies. I mean, 1 copy, so total 2. Then you the second cycle, same thing again repeats. Another cycle again repeats. So yes, you are doing temperature control. So this is done by thermocycler, which will do this temperature control automatically, right? And so you just put the sample, do the PCR, press the button, and you see the answer, right? So DNA is amplified. Okay? So this is a very, very basic, very, very important. PCR is a very, very important thing. Polymerase chain reaction. Any question? One can ask after n cycles, how many copies of DNA will be? Any idea? How many copies will be there? You can see that answer is if in another cycle, 2 days n copies are there. Right? So, I mean, you could have read it, but anyway. You can see why it will be there, right? For each cycle, how many copies will be there, right? Got it? So this is the idea of PCR. Very, very beautiful thing. Second concept. There is animation also you can watch the whole process. This PPT anyway is there, right? In lecture folder you can watch. Another system is required for measuring the length of the DNA strand. Length of the DNA strand. That is done using a reference point. So if you look at this here, this whole system is designed. Again, I would say that there is a link which I'll show you where you can actually design this system by hand virtually. You put a comb, you put all the material and so on and so forth, then you can design this thing experimentally. And then what happens? Because of the charge, so basically, if there are DNA strands of different size in the sample, they, you put the charge, so they will actually arrange as per the length of the DNA molecule. Okay, so they will move. Okay, and which one will move slow, which one will move fast, shorter strand and the longer strand. So you can arrange them. Okay, and then you can read the PCR. So, read the gel electrophoresis. All these things were used, by the way, in the DNA computing. It's a very, very standard thing for measuring the length of the DNA molecule. I'm not describing it in detail, but because I don't need it at the moment. But PCR is very, very important, right? So here is a homework that you go to this link and please do this experiment of gel electrophoresis, right? You can do the animation, virtual thing. You don't have to do anything. It will take hardly, it will require 10 minutes for you to do this thing, right? Okay. So there are 3 experiments I am giving. First experiment is gel electrophoresis. This is number 1. Number 2, extract your DNA. Again, there is how to extract your DNA at home. At this link, you can see. I think, how many of you tried extracting DNA? Anyone? So that is something you can do. You will really get a physical attraction towards DNA, right? So you can extract it from strawberry, you can extract it from banana, or you can extract it from whatever source, or on your own from the cheek, right? See, I mean, look at your own DNA, and then you see how to sequence that DNA. So basically, there are sequencers which can read that data and will give you maybe a CD or pen drive with all your DNA. I mean, if you don't want, I mean, to do it yourself, you can actually use this company, right? As I mentioned, in India, this company, Genome is providing this service. You send the, in US, 23andMe is providing this service. There are many other companies which are providing this service, right? So they can give you your DNA back. Okay, so by the way, I have covered many things in biology in 3 lectures. I would say that please do register for this course. It's about 16 weeks, 5 to 10 hours per week. So how many weeks are there? I mean, so how many hours? This is a must course for every person, you know, in computer science. This course is taught by Eric Lander. It is available on EDX. It's free. The only thing is that if you want to get a certificate, you know, you need to pay some fees. And many times, actually, if you write that you are below certain level of income, you can get it free also. For that, what they are going to say that you please look at 1 or 2 lectures and send us a summary that how it is benefiting you, and they can waive the fees. So you can get the certificate in any case, right? So this certificate, this course is by Eric Lander. Eric Lander, I mentioned, he was the person who did remarkable things. So he's a coding theorist. He did his PhD in coding theory. Now he's a professor of mathematics, computer science, and biology at MIT. He's also director of a few institutes right there, doing a lot of things actually. He was also in the US Council of Science and Technology to the president. He was, I think, vice chairman or chairman, I don't remember, but he was a member there anyway. So I think he visited India also sometime back, a couple of years back. Okay, so I think please do watch this this course. Please do this. It's very, very important as a computer scientist, right, to look at this course. You will learn many things. I have gave you some idea, right? But you will enjoy biology. Okay. So next time I will start the Fontaine codex and other 2 codex, or maybe the synthesis machine. We'll see. Something will come. I have to still prepare. Okay. Thank you.